All right, good afternoon, everyone. So I would like to skip the first part of the talk, which has been given by Nicola in the morning, and just jump to the details. All right. As you have noticed in the morning, our project is tackling with the issue of mental health and developing advanced AI and machine learning algorithms that can improve the prediction and the classification of psychosis, which is a complex task. And in collaboration with our NTU partners, we have received multimodal sets of data that includes genetic information, metabolomics, neuroimaging data, cognitive assessment. And as you can see, the nature of the data is complex because they sit in different domains in terms of time and space. So we have received this data from a cohort of patients who have been identified by ultra-high risk of psychosis. We have identified 3 main modules in this project. The first module was related to data generation, data pre-processing, and data engineering, which has been mainly led by our Singapore partner. So the second module is about developing algorithms that includes algorithms for a better feature selection, better data infusion and integration, and better learning algorithms to provide diagnostic and prognostic with high accuracy, as well as providing explainability in the field of AI. So this is something that is led by both groups in collaboration. Based on our developed algorithms, we have actually made a software, and the next step is deployment of our software and its clinical translation. All the algorithms that we are developing, they are in the field of personalized modeling for patients. That means we are creating a computational model for every patient using not the global data that we have, but using the most relevant samples to that patient, both in terms of the samples and in terms of the features that we are using in that data set. And of course, the aim is actually enhance the accuracy of the model, as well as enhance the model interpretability. And as I mentioned, we are dealing with multiple sets of data, such as genetic data, cognitive and clinical assessments. Some of them, they are just static data, and some of them, like gene expression data from more than 35,000 genes only in one data set. They have been collected longitudinally over 2 years through multiple follow-ups from our patients. And because we are dealing with multimodal sets of data, we have developed our algorithms in 2 main categories. One of them was related to singular modality, and the other one multimodality. For singular modalities, the intention was actually identifying potential biomarkers that are sitting in different sets of data, such as in genetic data, we may have significant genes that can be used to improve the accuracy of prediction instead of using the whole 35,000 or even more number of genes. So one intention was developing methods that can identify biomarkers for prediction as gene signatures, and also modeling longitudinal data through machine learning approach. In the field of feature selections, we actually developed new approach, which is based on selecting important features through multiple layer of feature selections that are actually getting benefit from different feature selection techniques and approach, such as filtering, wrapping, stacking technique. And the intention is actually to combine them together through multiple layers and achieve the best possible biomarkers. So I'm not going into the details, just providing you one example that shows the feature selection has been promising and could actually improve the accuracy of prediction of psychosis in just one of the data set that we were dealing with, like genetic data, compared to the other feature selection techniques. And we also have been able to identify a number of top genes. And we are at the level of IP development trying to protect those genes and validate them. And so this research has been actually submitted, and it's already under review by Nature Machine Intelligence, and one of our PhD students, brilliant PhD student, Balkaran, has been leading this, and he's already here. Sorry, Suga. So this is by Balkaran. And another model that we have implemented in singular modality was actually trying to improve the explainability and interpretability of the model. What we have done here, we use personalized modeling, and we develop personalized constant neurofuzzy inference, try to make the machine understand what type of interaction between multiple genes led the machine to make a certain decision. So in this case, our fuzzy inference learning could provide rules, fuzzy rules, to say if there is specific interaction between a specific number of genes as biomarkers, this person is going to develop psychosis or not in near future. And as I mentioned, we also develop our software. We have given just draft name, initial name, Neurogene, still brainstorming the name. This fuzzy rule extraction has been applied to genetic data set as well. That could improve the accuracy of prediction, as well as we could see a number of rules, fuzzy rules, were extracted across the top genes to distinguish between a control group and people with psychosis. This is also submitted and it's under review in Nature Scientific Report, and it's also led by one of our PhD students, Balkaran is here. Another set of data that we were dealing with that was related to longitudinal modeling of cognitive assessment and behavior data that were collected over time. So we actually use deep learning, and also we use spiking neural networks to model the trend of changes in psychological and cognitive assessment over years, and see if we can find specific interaction between cognitive assessment over years, and we can use that pattern to predict the risk of psychosis. And this research is also under review in Nature Schizophrenia, was led by our postdoc, Zohre. She's based at the University of Auckland. And I talk about multimodality as well. And as I mentioned, because we deal with multiple sets of data, we have defined two approaches, and for each approach we develop multiple algorithms. So the first approach is early fusion of different data modalities prior to passing the significant features, combination of features, into machine learning to extract patterns. And the second one is a late fusion, versus we actually in here, we pass our features to machine learning, and during the learning process, the features will be interacted, and we select abstraction and significant features. Fusion was promising, the technique, and has shown by applying early fusion, we could improve the prediction accuracy compared to just using different modalities of the data separately, versus when we use integration techniques. And this is our methodology for integrating different data modalities at the learning phase. We are still developing this to be applied onto different sets of data, and let the features to be trained in parallel using different neural networks, such as spiking neural network, LSTM, and deep learning. Another thing I would like to mention is clinical assessment tool, which is under development and has been led by Samuel, who is a PhD student in NTU and a visiting researcher here at the moment with us, and he's here. This clinical assessment tool was developed as a kind of psychosis risk calculator, and to actually to use temporal data and predict who is going to have psychosis with a certain percentage and in what time. So this is just an example of 6 months ahead of prediction of risk of psychosis for only one input data as the testing, and you can see for this individual there is a 10% of psychosis within 6 months. And yeah, so this is just a wrap of that. Only in this year we have actually submitted and published a number of papers in reputable journals. Okay, in reputable journals, and we have presented also for knowledge dissemination aspect of our work, and there were 2 posters from our group already here. And yeah, so we have also received a number of awards as a recognition of our achievement in this project. Thank you very much, Jacinta.